for them in the US just recently, but you hear a bit more from him, and then I'll introduce the next speaker afterwards. Could you please play the video? So this is what we launched just this last spring in the US. So a lot of exciting things are happening. So you see NFL players and some famous Megatron, a lot of partners that we've partnered up in this. Please play the video now. Thank you. Invested in a farm in Cullinan in Pretoria East, where I've been doing livestock farming and cropping as well. And then, of course, the opportunity for the licenses for the medical cannabis came along, and I was one of the first 3 that were issued in the Republic of South Africa. It hasn't been easy. It comes with its own cost. What was interesting for me today was after hearing the gentleman from agriculture saying that they're going to do away with some of the conditions like your police checks, your camera systems. Yes, I do agree with that, and thank you so much. At least you make it easier for others as well, because it's actually very capital intensive. So with some of those changes, we welcome them a lot in the industry as industry players, and we're also looking at collaborative partners. If we can get, we can also offer off-takes as well. We've brought in a machine for extraction as well that we're going to be setting up, and also Afrimat have got a decotylation plant which they can get all the hemp stocks to process into hempcrete blocks, which will be for your building materials and the like. So what we need is a centralized system where everybody's got access. I've been talking to the minister earlier and also to the Department of Agriculture to say we need to align ourselves. It appears like we're all working in silos currently, so that's why we're not having a lot of progress. So we need to all come together, but more, thank you. But more importantly, the education aspect of it, because we are saying let's create wealth for the herbalists and health for the people. So how do we bring you on board, get you to Harvard, get you to Oxford, get you to Stellenbosch, and then look at your products and say, right, okay, what are the safety measures, how do we store them, and so forth, and help you with it. But how do we protect your IP? This is where we need a platform where we can protect your IP. So we've created a session called phytomedicines, medicines from plants, and medicinal cannabis. There's been a buzz about cannabis, but there is more to it than the cannabis. We're also looking at the phytomedicines, which for us and for myself really think that that is the way to go, because we need to start moving away from using a lot of opioids and then start tapping into our own indigenous plants. And there's so much knowledge that we can also extrapolate from our indigenous our herbalists and so forth, but we are not, I've summed us Africans as victims of our own success. There are only 2 things that are missing: organization and management. If we can organize ourselves and manage ourselves, I think we can be the winners, really. But unfortunately, we've not been able to do that. But I'm hoping from this day forth, we'll be able to put something together. I'll just check if they're ready with the audio. If you're ready, please can you project it so that at least we can move forward. That says it all. I'm sure if you looked at it, you can see what the message is about. It's about education. The stigma around this, we need to like do away with it. So let's educate ourselves, and especially on the continent, you find we've only got a handful of countries that have legalized the medicinal cannabis, and there's still the stigma. So the education is very important. I'm not going to take too much time. I would like to get Dr. Nge, who is sitting in the US, to give us his presentation. Dr. Nge, please can you start your presentation? I would appreciate it. And this is how far we've gone with collaborative partners. These are people that we've collaborated with and now going the clinical research studies, which has been approved by the FDA, which we launched just May gone in University of Pennsylvania in the US. Please, Dr. Nge, go ahead. Thank you. Just a quick note here. Women in Leadership is now in Hall 22 DEF. Women in Leadership session, it was going to be in this room, but it's now going to be in Hall 22 DEF, and it's happening now for those who are wanting to go, but I would love to keep you here if you're able to stay in. Thank you, women. Dr. Nge, please go ahead. Whilst we're waiting for him to stream in, we can start preparing your questions for us for later. Then we're happy to have your questions as well. But I'll just get a message across to them to try and connect to the link. If you just give me 1 second, I'll be able to get them connected for you. Thank you.
Good day, Doc. Can you just try and talk again? Yeah, hello. We can hear you, Doc. Okay. Please proceed.
Right. Thank you so much, Dr. Nge and Flavorcure Biotechnology, and congratulations for the FDA approval for the clinical research. I think it deserves another round of applause. Right, I will now ask Dr. Sandra from Panama just to talk about the whole endocannabinoidal system and innovations and clinical use of medicinal cannabis. And I have brought these people on this program so that we can start getting collaborative partners. Dr. Sandra, could you please go ahead? And thank you so much. Dr. Sandra is from Panama. Thank you, Dr. Sandra. Thank you so much for that. I mean, you've heard it for yourselves from a clinical practitioner and also a member of the International Phytomedicine Institute, where you can see El Paso Farms is the farm that I own. So you can see the giants we've got, your IBM, your Harvard, your UPenn, and we are looking at making a serious footprint in Africa, on the continent, because this is where it starts. And if we cannot start it from South Africa, I don't know where we can start it, maybe half of Africa or the top, but I believe that if we start from the bottom, and then we can cover the whole continent. Thank you so much once again, Sandra and Dr. Nge, and also I just want to say thank you so much to Professor Wungwa from Harvard, who started the whole movement at Harvard, the Global Health Catalyst, which is now we've now got that International Phytomedicine Institute, where I'm looking forward to having a lot of you on board as well. I'll let Dr. Blessed introduce the next speaker. Thank you so much once again.
Thank you. Thank you, Brailin. We'll continue with the next presentation, which is from Professor Matsabisa. Please, your slide is on. Prof, I'm not going to do a lot of introduction. Everybody here knows you. You have the floor.
Thank you so much, Doc, and thanks to everybody. I think in the next 10 minutes, I would like to start off by some anecdotal evidences in terms of what we have been picking up and seeing. One is an old lady that has arthritis. The lady could not even use their fingers. And then we talk about someone who has got chronic back pain because of, I think, a spinal nerve that's being pinched. And then we start talking about aspects of memory loss and generalized pain. And then how do we explain that? And I really want to say thanks to, I think, Nge for talking about non-cannabinoid compounds from cannabis. And I want then today to talk to you about some of the work that we do in terms of cannabis. But more interestingly, my interest is not on the cannabis, CBD, THC. It's on the combination of those. This morning, I think we're talking about local land races. I work specifically on the samples that you'd find, I think, somewhere on the streets. I think, I work with, I think, Mimi, I can see her there, from Velcom, and collecting all these samples and trying to analyze them to make scientific understanding in terms of why users would be saying, the Buddha cheese is the best performing, that is the best performing. So let's see why. These are some of the extracts that we have done. We do cold or frozen ethanol extract, simply because ethanol is cheap. Ethanol is safe in terms of residual solvents. And then our interest was to look at those combinations of the different cannabinoids and begin to say in what ratios would those cannabinoids be for a particular disease. So you try and develop a disease based on the cannabinoids. We started off, I think, with those 4 samples from different plants, and we analyzed them to say what were the cannabinoids in there. And how would these plants justify the arthritis that we have seen on the extracts? How did they justify the pain that we have been reported to, that I took this, sample and I got the relief of pain. I took this sample and I slept like a baby. And now we're beginning to understand what's happening there. I'll not go through this, and again this morning we've been hearing about THC levels of 0.2, 1%. If a plant produces, and I'm going to show you that I think some of the plants that we see on the streets can produce THC of up to 15%. So what do you do with that? Because our understanding is the determination of the concentrations of the cannabinoids should be based on science, should actually be based on their efficacy. So these are some of, I think, the quantifications that we have done on the samples that I've shown you, and you could see that the cannabinoids, we looked at CBD, CBC, and all these different things, including THC, and you could see a range in terms of concentrations. I think those are in micrograms per ml. But what were the ratios in the different samples? Because that's where my interest is, to say if this sample gives you an anti-arthritic activity, what are the ratios of those cannabinoids? So I'll run through these, and these were just giving us the different samples, but this is where the important, I think, slide is, where we looked at and say, fine, how are these distributed in those? I don't know why this thing keeps, blinking, but we then looked at, for each sample that had a known pharmacological activity, what were the ratios of those cannabinoids? Now that we know what those ratios were, how then do we optimize them for a particular disease condition? So the work that I think we are doing ranges from pain management to Alzheimer's disease, looking at arthritis, looking at diabetes, because we know that different combinations of the different cannabinoids would then be specific for a different disease. And so this is some of, I think, the hydrogels that we are producing for slow release, for pain management, for slow release for wound healing. But also the work that we are doing in terms of pain and Alzheimer. So if you were to look at, I think, the top, row, the top row are the cannabinoids that we have found commonly in those samples. And these are the samples that you find on the streets. But if you then go to the next row gives you the combination of cannabinoids to treat a particular disease. So now you look at Alzheimer's disease. We know that for Alzheimer's disease, you need that combination. The question is, in what ratio are those cannabinoids? And you could immediately see that, in fact, those samples that we have been collecting do have the same cannabinoids that we know are good for a particular disease. You look at, pain similarly, and then the red cannabinoids are those cannabinoids we have not picked up. But I will give you the relationship. So what we have now seen was, in fact, then those cannabinoids could explain why the people that were using those extracts are now saying, I got relief for my arthritis, I've got relief for my pain. Now, CBV, it's one of those general anti-cannabinoids. It has the same function as your anandromide, which is your endogenous cannabinoid that you find in your body. So its function is virtually the same, and its function is virtually the same as CBV. So that we have CBV and CBVC, it's giving the same activity. THCV, it's similar to THC, except that THCV has a propyl group rather than a pentyl group, and therefore THCV does not give you a high, but it does have the same medicinal activity. So we're beginning to understand why different extracts or why extracts of the same could have different pharmacological activities. And where we are now is then to work out the exact ratios, even though we know from our previous slide, I think the slide that I showed you, I think it is that slide, where we could now determine in those extracts what were the ratios that were in those plants. I think this is the slide that I was looking for. So we know what those ratios are, and therefore one could then develop a product based on specific ratios for a specific disease. So I think in conclusion, one wants then to say, one could then move forward to begin to say the final concentrations of either your THC should be determined by the pharmacological activity. I think regulations should then take a clue in terms of what is the concentration of THC in the final product, not per se in the plant itself. That's the group which I really want to thank, doing all the hard work. I'm just pushing them from behind. Thank you so much.
Thank you, Prof. I told you it doesn't need any introduction. He's told us what is happening. This is happening here in South Africa also. We heard what is happening with our colleagues in the US and Panama. So we're also doing it here in South Africa. I wish we could just be working together to come up with innovative products. I'll call my next speaker, Dr. Mofokeng. He's going to talk about the seeds that we've been hearing about and also some other research that they've been doing at the ARC. Thank you.
Good afternoon, ladies and gentlemen. Thank you, Dr. Okole and colleagues. I'll be giving a short presentation on the agriculture work that we do on cannabis in support of other stakeholders. I don't know. So as the Agricultural Research Council, our mandate is basically to conduct research on various plants. And we have the Medicinal and Industrial Crops Research Division that has got cannabis as one of the crops that we are working on. Colleagues, you will remember that previous speakers spoke about the number of permit applicants that or people who have been awarded permits to start growing and cultivating hemp. Oh, it's there. To start growing hemp and also cannabis. So the value chain, I think, is very straightforward, as like in any other crop. First, access to genetic material, propagation and cultivation up until you have a product on the shelf. And okay, and in all these areas, research can play a critical role. One, in the preservation of genetic resources and protection of the intellectual property or knowledge related to the genetic material, value addition until you have a product on the shelf. Now, as the ARC, I think it has been mentioned previously that we have been working on hemp for a number of years. I think for about 20 years, where we had to procure and get or import material from other countries, start growing them, evaluating them, start the breeding program, and we are happy to say currently we have 2 local cultivars that are in the process of being registered. And from the R&D program, we have also developed a hemp cultivation guideline for South African growers. This guideline is continuously updated as we continue with research. Our current research is now focusing on development of varieties, as well as conducting adaptation trials in various areas of South Africa, just to ensure that there is sustainable production with minimum production costs. The story of SA1 and SA Hemp, SA1 and SA2, I think, is known. And interesting is that it performed very well compared to other hemp varieties that were imported. We could see the yield was in the region of 8 to 12 tons, the fiber percentage, 28 to 32%. So the breeding program that is in the ARC is aimed at ensuring that we have genetic variability of the locally identified land races as well as the imported varieties. We aim to maintain the germplasm, also to use different techniques to ensure that we have a database, DNA database of the land races. This is just our gene bank facilities where we collect various accessions for maintenance, and we keep them for a number of years. We monitor the seed viability, and we rejuvenate the seed accessions. So the rejuvenation is basically because we know that the seeds will take 1 to 3 years. After that, the viability is very low. So we have to plant outside, rejuvenate our accessions. Yes, it was mentioned again earlier this morning that we have done some evaluation on the genetics of various accessions. I think the earlier speaker spoke about Durban poison, where 2 sources seem to be different. So we are using different techniques to do that in the ARC. So once we collect the local germplasm, we also try to do the molecular markers and the genetic identification so that we have a database of our local land races. And that will basically help us to protect the indigenous knowledge around the genetic resources. So we go further to use the different techniques. So other techniques, we just look at the genetic fingerprinting of the resources or the genetic material. We go further, which is the new project that we are working on with our ARC partners, into looking at the makeup of the genetics themselves, the building blocks of the genetics, to further identify the local land races. And they use the long-read sequencing of local varieties. And this work is done by our colleagues in the ARC Biotechnology Research Unit. For the past season, we have been evaluating a number of cultivars that we have in Gauteng, and we will take the project further to evaluate in other non-traditional provinces or areas in South Africa. I think there was a question to say that if people are getting permits in different provinces, which cultivars will they grow there? We are trying to identify different cultivars that can be suited for different agroclimatical regions of South Africa, so that we can then advise the growers, the permit holders, and the farmers. Still to be finalized, I think I've highlighted that because I know that some of our collaborators and partners will be here. We have identified a number of areas in different areas in provinces of South Africa where we would like to continue with the cultivar evaluation trials. I think it is now known that if you want to grow hemp for fiber, you have to plant them closer, but if you want to grow them for seeds and flowers, you grow them wider. But there are other different agronomic practices that are involved. So we are also looking into pests and diseases around different cultivars of cannabis. We're also looking at irrigation and nutrient management to improve the yield. And once that information has been generated, remember, colleagues, we are trying to support the department, so we have had training with the different officials from different provinces, Department of Agriculture as well as the National Office. They visited our trials and we have given them training. This training, in time to come, it will also be taken to the growers, the farmers, and the permit holders in terms of the different agronomic practices that can be used to grow cannabis. And we are talking both hemp and medicinal cannabis. We look into different production systems. Do you want to grow them under drip irrigation, which is a bit more expensive, versus under rain-fed conditions? We look at issues of under protection versus just under your normal shade netting structures. In conclusion, ladies and gentlemen, the slides are disappointing me, but I hope it's showing now. We can say that for the collection of accessions, that is continuous work. We are collaborating with different institutions, universities, other science councils, knowledge holders, and we are making a plea that we would like to collaborate further. If there are knowledge holders who have genetic material and would like to collaborate with us, there are processes to ensure that we protect your knowledge, we don't take advantage of you. We have developed a cultivation guideline, which we will continue to update. And there are 2 cultivars that are in the process of being registered. We hope that soon the process will be finalized and they will be available. In the ARC, we are also busy with procuring equipment that will assist the growers in terms of quality control, and that laboratory will be accredited, because some of the growers or the permit holders would ask us, you saying that we should do this test, but where do we do the tests? Okay. And also the last part, which I've indicated, is the training to support the government departments, as well as the permit holders, the growers, and the knowledge holders. We would like to thank all the collaborators who have worked with us before, the knowledge holders who have worked with us before and who will continue working with us, and the different government departments for the funding. Thank you, ladies and gentlemen.
Thank you, Dr. Mofokeng. Any burning questions for Prof. and Dr. Mofokeng? I see everybody wants to go for lunch. Oh, there are 2 questions. Okay, we'll take. I don't think we have the privilege to have a microphone. If your voice is loud enough, yeah.
Good day again. I'm Timo Hase from Department of Agriculture. Mine goes to Prof. Masalis. I want to know if from all the products that have been developed, have you ever tried to register any of them with SAPRA?
I didn't get the question. Can you just repeat the question so that, Prof, I think it's for you. Yeah, registered.
I want to know if any of the products that you have been developed or working on, have you ever tried to register them? Are they registered? Because there's a medicinal claim that is attached to them. So that's what I want to establish.
Okay. Thanks, Cecil. The University of the Free State does have a research permit, from SAPRA to do the research. And the process for us and the intention is to get products onto the market. But also I think the intention is also to influence policy change. That's why we started asking about these concentrations of THCs, because a concentration is determined by science in the lab, and whether it works or not. So we have not got to the stage where now we are saying we are sending the product to SAPRA for registration. We are still at the pre-clinical stages. But eventually and ultimately is to get those products out onto the market.
behind there. Yeah.
Over there.
Why is the guy with the mic?
There. And that's for you.
Thank you very much for the insightful presentations. My name is Dr. Musengi from Harare Institute of Technology. My question is directed to Dr. Mofokeng. I would like to find out how far he has gone with clonal propagation. Have there been any success stories in bringing clones to the market? Thank you.
So the question is, how far have we gone with clonal propagation? Yes, we have used the tissue culture technique to propagate some of the material that we have, and it's successful. But it's a bit expensive to take tissue culture material to the growers and the farmers. That is why we have the seed multiplication program. At least it's a bit cheaper. But the advantage of the clonal propagation is that you know that you have true-to-type material. So with our seed multiplication, we are trying to do it indoors so that we don't have influence of other genetic material. Thank you.
Thank you. Any question from the ladies? Well, for the tissue culture, you can consult me. I will make it cheaper. Just advertising my service. I'm an expert in tissue culture. Colleagues, thanks very much for being patient. I'll all invite you for lunch, and it's been a great session. Let's give a round of applause to all the speakers and the patient listeners. Thank you. The next session is going to be on, you want to do it? There's one more session on industrialization, which will start at 3 o'clock. We're going to announce the venue. Unfortunately, the venue is not on the programs, but there is a session that you're going to see what we're doing here in South Africa on cannabis and hemp, and it's going to involve SMMEs presenting the challenges and the products that they've produced, and also on the hemp side, what we're doing in products that have gone into the industry. So it's more on commercialization and industrialization. Thank you very much. Thank you, colleagues. Thank you, madam.
Thank you so much.
Thank you. Thank you, Doc.
Eventually I'll come to the stage. Let's get rid of that presentation.
Let's go do it. Let's go do it.
We've got a permit for the ARC.